Hi guys, I'm making a video for our Lab Practical 1 just to review so you'll know what to study. So remember I said that you needed to know all the phases of mitosis for the animal cells and the plant cells. So make sure that you can recognize prophase, metaphase, anaphase, and telophase. Okay, you'll have the animal cell, that'll be the first 4 questions, and then you'll have the plant cell. So here's the plant cell down here. Prophase, metaphase, anaphase, and telophase. Okay, so make sure that you can identify those, and that'll be the plant cell will be the next 4 questions. So make sure that you can look in the microscope and it's already going to be set up, and you just have to be able to tell me which phase it is. So make sure you can recognize that. And I'm not going to have the phases on there more than once. Like, for example, for the animal cell, I'll only have prophase, metaphase, anaphase, telophase once. I'm not going to have 2 of them that are telophase and then 1 that's prophase and metaphase and leave out anaphase. I won't do that. Okay, so keep that in mind. You'll also need to be able to recognize the cleavage furrow. Let me grab that poster again. Okay, so the cleavage furrow in the animal cell, and then in the plant cell you have the cell plate. So I'm going to have 2 questions about those where you have to recognize those. And then we have the models. You're going to have to identify one of the organelles on the model. So let's go over them really quickly. We have the nucleus with the nucleolus in the middle. We have the centrosome with the centrioles, Golgi apparatus, mitochondria. This right here that's green and blue with the white dots on it, that is endoplasmic reticulum, and it's the rough endoplasmic reticulum. The part that's in here that's just the dark blue without the green and the white dots, that's the smooth ER. Alright, so plant cell. Here we go. We have the nucleus with the nucleolus. We have the central vacuole, chloroplast. The rough endoplasmic reticulum is the one that has the green with the white dots on it, and then the blue, dark blue part sticking out, that's going to be the smooth endoplasmic reticulum. Here's the Golgi apparatus up here. And then also remember that the plant cell has the cell wall on the outside. Okay. You also want to make sure that you're studying some of the review questions and looking through some of what we did in the lab. You want to make sure that you know how to tell me what the total magnification is. So if I say, what is the total magnification with the low power, then you know that the ocular lens is 10x, the low power is 10x, so 10 times 10 is 100x. Remember, scanning is 40x, low is 100x, and high is 400x. You'll also need to be able to identify the parts of the microscope, so I'll have the microscopes set out, and it'll have a sticky label on the part that you have to identify. And remember, we looked at the prokaryotic cells and then eukaryotic cells in the cell structure and function lab. We looked at the kidney cell. In that cell, it was stained pink, and the nucleus in each cell was purple. So be ready to identify the nucleus in the kidney cell and be able to tell that that's a eukaryotic cell because it has a nucleus. And then the prokaryotic cells are the bacteria cells, and they don't have a nucleus. So be able to recognize that. If I have the bacteria on there, you'd be able to recognize that they're prokaryotes. Also know things just that we studied in lab, like some of the functions of the parts of the microscopes, the functions of the organelles, let me think. I already set up the test, or I'd be looking at it telling you what's on there. So I'm just trying to remember what did I do when I set it up. Also, remember we worked with the pop beads for meiosis, and I'll have pop beads on there, pop bead chromosomes already set up, and I'm going to ask you to identify the parts like the centromere, which is the magnet that holds the two strands together. The two strands are called sister chromatids. But then if you're looking at a homologous pair and you're talking about a sister chromatid, you know, a strand in one, and then comparing it to the strand in the other chromosome, remember those are called non-sister chromatids. I'm going to ask you to identify all of those things. There's that one question that's on the test that was in one of our labs that was kind of tricky for a lot of people. It said if there's 13 pairs of chromosomes in a sperm cell, then how many chromosomes, or if there's 13 pairs of chromosomes in the parent cell, how many chromosomes will be in the sperm cell? And so the tricky thing was that it said 13 pairs. That means that there's 26 chromosomes. So in a sperm cell, there would be 13 chromosomes because the diploid number is 26, but the haploid number would be 13, so the sperm cell would have 13 chromosomes. That question will be on there. In meiosis, I had those microscope slides set up where you could see the different stages of meiosis, and I'm going to have a question on there. It's going to be a bonus question, and it's going to be showing you one of those slides and asking you if this is a representation of mitosis or meiosis, and how can you tell. So it's going to be meiosis, but make sure that look over the steps of meiosis so that whenever to answer the second part of the question, how can you tell that this is meiosis, you'll be able to tell. And I believe that's it. There's going to be 32 questions on the test plus one bonus question, but the one bonus question actually has the two parts, so it's like two bonus questions. And I believe that's all. Please let me know if you have any questions.